Thank you. It's great to be here. So I'm going to talk a bit today about some of the work we're doing in rare disease gene discovery, as well as genomic medicine implementation. Yesterday in the VAR-I COSI, I spoke more about clinical variant interpretation and shared my slides. If anybody wants that talk, I have a URL link for that too. But I'm going to talk more about rare disease data, genomic data discovery. A copy of these slides are using that QR code, and which I'll put on the last slide as well if anybody would like a copy of the slides. I do have one disclosure. I receive research funding support from Microsoft. So in terms of discovering the causes of rare disease, just under about 5,000 disease or genes have been implicated in at least one disease. Some genes are actually implicated in many different diseases, but we're at about 5,000. The challenge is that when we sequence a patient that we suspect has a genetic disorder, over half of the time we don't find an answer. So we're clearly missing many of the causes of rare disease. And there's evidence that at least 5, if not 10,000 more genes yet to be discovered for monogenic disease. That's using constraint data from our Nomad database, as well as animal model data that suggests many of these genes are implicated in phenotypes. But I also think that even if there's another 5 or 10,000 genes to be discovered, there's probably a good percentage of the lack of discoveries that are understanding variation in known genes that we haven't yet been able to interpret the variants for. So there's a lot of work in this space that needs to be done. We have a large team at the Broad Institute in our Center for Mendelian Genomics that's been tackling this area. The center is co-led by Anna Donaleria, Mike Tchaikovsky, and myself. And we've been really working to develop both collaborations and new methods and tools for analysis. And I'll tell you about some of those approaches. This is a picture showing a lot of the academic partnerships. So we rely on many researchers that work in specific disease areas to recruit patients in those areas. And then we have a direct recruitment effort through what we call the Rare Genomes Project, where we directly enroll patients who are undiagnosed. Most of them have had clinical testing that was negative. And then we can apply new technologies and techniques. And we also partner with patient organizations. And this has led to us being able to amass over 20,000 samples from all around the world. We also more recently have been trying to tackle the challenge of the lack of underrepresented individuals in some of our data sets. And so we've been really very specifically thinking and trying different methods to bring better representation. For example, what we used to rely on is have the physicians tell patients about our study and then have them contact us. Well, when you're a busy family, it may not rise to the top. So now those physicians give us a referral form. We reach out to the patients and we ask, can we help you? Can we help you navigate the consent form? Can we do anything for you? We also now have hired staff that speak multiple languages and have translation services. We have staff that work on nights and weekends, so we accommodate the schedules of families that can't call during business hours. We also work with Quest to have mobile phlebotomy, so we send blood draw Quest individuals to their houses on weekends to get blood samples if they can't come in during business hours. And these are just many examples of how we can improve our enrollment, and that's starting to increase the numbers in terms of the diversity of our data set. The other way that we democratize and distribute analysis to families is actually by simply making all of the tools that we build open source and freely accessible. So we built the Seeker Genomic Analysis platform, and it's a ability to load any sort of exome or genomic DNA sequence and then be able to have all of the variants called and analyses done. So we've been incorporating many features into the software over the years. We have RNA-seq data. We're working on adding long-read data to that. We have installed it on the Anvil platform, the cloud-based NHGRI-funded platform where there's lots of other tools and data sets. So it's there, it's FSMA compliant, and people can just use it there and upload their data. or you can install it as in a local instance. And a number of our collaborators have installed it locally so they can manage it in their own local cloud or on-prem environments. So to date, we have over 2,000 people from over 100 countries that are using this for analysis. This is just a video or a visual of the Anvil platform where we've installed it. But just as an example of that, having put this software there and put all of our data sets there, Aaron Quinlan, who was developing a computational algorithm for structural variant detection, ran that on the Anvil platform on our data and actually made a diagnosis in one of our cases with an upstream structural variant. So it's really nice to have a place where all of the data and tools can be shared and be more accessible to the community. Now, if we look at the data sets that we've been analyzing, and we have traditionally run exome sequencing on most of the large data sets from our collaborators. Through funding from Illumina, we received all of our Rare Genomes Project samples have gone directly to genome sequencing. For our most interesting genetic cases that have gone on had exome, but then were negative, we've pushed then to genome. So we have a lot of different sort of solve rates depending on what technology, genome versus exome, and whether they're fully solved or they have candidates. But if we just look overall on the right side of the slide, we have solved about 35% of cases where it's a clear solve, but about 45% have been negative, and then another 20% inconclusive due to some sort of variant or candidate that we haven't been able to securely implicate. So I'm going to kind of walk through some of the reasons that we don't solve cases and how we're tackling those things. So in terms of the negative cases, there's usually two main reasons they're negative. I should say there's a third reason is that they're not genetic, but assuming a lot of these that we suspect are likely genetic, it could be that the variant is simply missing from the sequencing. It's not there, so there's nothing to interpret. The obvious thing is when we've run exome, we then push to genome, and that will cover more variation. That's no surprise. One example of a really huge discovery pushing from exome to genome, and this was just published last week on July 11th, was the discovery that RNU4 to the small nuclear RNA actually is probably one of the single most common causes of neurodevelopmental disorders. And that is basically none of the exome data sets, the probes, included small nuclear RNAs in them. They're only looking mostly at coding sequence. And this, there is a single variant, an insertion of a T in this small nuclear RNA that is actually incredibly common. And not clear why it's so common. And it's actually all of the de novo variants are in females, which is the opposite of what you typically see. You typically see it in males. So it's a really interesting finding. We immediately had 8 cases in our cohort that were unnoticed. So this paper just came out and represents a huge added yield for neurodevelopmental disorders. We did just prior to that run a deeper analysis looking at what is the true added yield of genome over exome and really parsing that in terms of things that absolutely require genome, as opposed to if you just reanalyze data, you sometimes find things, but you could have found it by exome anyway. So we really were differentiating that. And what we found was that our genome analysis yielded an additional 8.3%. And that's largely drawing from the things that are in dark blue here, structural variants. Coding variants had poor coverage in exome. The deep intronic variants, often now detected by splice AI signals, which we found has been a pretty accurate detection of both splice disruption and creation of cryptic splice sites. And then triplet repeat expansions or tandem repeat expansions also. What I didn't add, because it happened afterwards, is all of the yield from the RNU4 cases, which would have increased the 8.3 up higher, given just how many of our cases had that. But that gives you a sense. This was just published in New England Journal of Medicine a couple months ago with a added cohort from Germany that had a similar rate that was found. Interesting story. New England Journal of Medicine rejected our paper, but then because we'd put it as a preprint, this group from Germany found our paper, said, hey, we have the exact same yield. Can we combine our data with yours and co-publish it? And we said, sure. And we went back to New England Journal of Medicine and said, our data has been validated with another cohort. And they said, okay, we'll accept it and we'll publish it. So keep in mind the benefit of preprints. All right, moving on. So in addition to just simply moving from exome to genome, even when you're doing genome sequencing, you aren't catching all of the variants because of some of the homologous regions that are difficult to catch with short read sequencing. So we've been working with long read sequencing, both ONT and PacBio. We've solved some cases. I will admit that most of the cases we've solved, we're ending up picking up things that could have been picked up by short read sequencing. We haven't seen a huge added yield yet from long read. That said, there isn't great population data yet generated from these regions that have been difficult to sequence. And so I think over time, as better tools and reference databases exist for these regions that are outside of the traditional short read sequencing accessible regions, we will start to yield more finds from this. But to date, we haven't seen a huge added yield from this. Where we have seen more larger yields is in the use of RNA-seq in combination with the whole genome data. And this is addressing the variants there in your data. You just can't interpret it and notice that it's causing disease. So just as an example, this is a patient with centronuclear myopathy with hypotonia, weakness, ptosis, and reflux. There's suspicion for myotubular myopathy that is typically due to MTM1 variants, but the exome sequencing was negative in this gene. We did a trio with RNA-seq on patient muscle tissue, and what we could quickly see was that the expression outlier most significant was in fact MTM1, the suspected cause. And then we could look at the splicing data and show altered splicing, skipping an exon in the gene. And it turns out if you look deeply into the deep intronic region, there was a line 1 element insertion in there. The mother was actually mosaic for this variant. So we could quickly identify the cause. And it's just an example of a pretty straightforward added yield by simply adding RNA-seq to your genome data. Now, this works great if you've got tissue that's relevant to the disease. So this is a muscle disease and we had muscle biopsy. It can be more challenging if your disease is due to tissues that are inaccessible, like a neurodevelopmental disorder, retinal, you can't take a biopsy of the eye or the brain easily. So in those cases, sometimes you get lucky and those genes are expressed ubiquitously and you can find altered transcription in blood or a fibroblast cell line, but other times they may not be expressed. So we tend to do proactively looking for RNA-seq data for any patients where we can get biopsies that are relevant to the tissue. And then we more proactively do it, or not proactively, but in follow-up do it for those diseases where we can't get tissue access, only when we have a splice AI signal that's interesting. And then we're willing to try it, even though we know we might not get enough transcription. But that has actually yielded a number of solves, even so. So here's another even more interesting case using RNA-seq. In this case, there were a ton of overexpressed and underexpressed genes. There wasn't a nice, clear outlier like that last case. And it turns out, and we actually looked in the introns of all these genes, we could not find anything that stood out as a variant that might be causal. Although interesting, if you look at retention of introns, which is an analysis you can do, this particular case stood way out compared to all of our other samples. And you can see here the inclusion of this intron, although sporadically including introns. Well, it turns out that the U12-dependent minor spliceosome, which splices about 0.5% of introns in the coding sequence, there's specific RNA, small nuclear RNAs that actually control that. And so there was ultimately identified a variant in this small nuclear RNA that only contributes to this minor spliceosome complex. And that's why these sporadic introns were being affected. And they're either looking like they're overexpressed because the added signal from the retained introns, or they're looking like they're underexpressed because some of these will rapidly degrade and show lower signals. So you're sort of artificially seeing this over and under expression when in fact it's actually due to this spliceosome defect. So those are just some examples, but overall, in looking across cases where we've had access to RNA-seq data from all these muscle disease cases, we found an added yield of 14% using this approach. So one of our higher sort of added yields. One of the even higher added yields is if you just go back and reanalyze your data. Turns out that's been our highest added yield of 17.7%. By the way, this 3.2 is looking overall at all cases and how many were solved by adding in things like RNA-seq. But the highest yield is actually reanalysis. And so in this case, you know, the variant's present but was overlooked, but over time, by bringing the ongoing new gene discoveries and variant classifications showing up in ClinVar, things like this, you can find new solves in your cases. So we've actually been developing an automated interpretation pipeline, because what we've had to do is every year have our analysts go back and reanalyze every case again. And that is really labor intensive to keep doing that every year, although we've been doing it and finding lots of new solves. But we wanted this to scale and be easier on the labor force. So we developed this pipeline. I don't have time to go into the details of it, but it's, you know, it's doing some things you'd anticipate, picking up new genes submitted to either OMIM or GenCC, as well as new variants submitted to ClinVar, things like that. We've validated with 2 cohorts. So this is a collaboration with Dan MacArthur's group in Australia using their CPG cohort. This is our Rare Genomes project. And we were able to recover 81% or 71% in the 2 cohorts of our solves in terms of our validation, which performed better than comparing that to Exomizer or Lyrical, which was done by the CPG group. So overall, it has pretty good specificity and time efficiency and sensitivity. When we run these now on unsolved cases, we see about 1 candidate per 200 cases per month. And it's really important that these tools don't just keep throwing tons of variants at you, because that's manual to go analyze everything it's throwing at you. And this is actually before we put in a phenotype matching filter, so that we would not be shown lots of things that have nothing to do with the patient's phenotype. So it's actually even more specific now. So we're continuing to evolve this automated pipeline, and we're going to actually now utilize it in a project that we've been starting, funded by the Anvil project, where we're basically working to bring in unsolved clinical testing exome and genome data sets. So we started working with GeneDx and have now a pipeline in place so that for any patient where the physician has consented them for reanalysis or any sort of research that includes reanalysis, we will then, on their behalf, bring the data from GeneDx onto the Anvil platform, create a bucket for them to store their data, and then they can analyze it using our Seeker platform for free. And then we will also run the AIP tool on it. And that'll just be continually running on their data. And it will, we haven't set this up quite yet, but it will be then sending them emails if a new finding is there. And then they can go visualize it in Seeker and then, make sure it's a phenotype match and all the things. And our team will be there to assist as well. So we really hope that this pipeline really facilitates getting a lot of these unsolved cases that have been sent for clinical testing that never see the light of day after they're negative, and really bring them into a pipeline to enable this sort of ongoing reanalysis, as well as access to other research analyses as the patients and physicians wish. So that's a project that we're just getting off the ground now. And then last is this bottom pipeline. So we actually have identified a variant. It's a candidate of some sort. It might be in a novel gene that's never been implicated in disease, or it might be a VUS in a known gene. These are annoying, but one of the strategies that we've been doing over many years now is to facilitate what we call genomic matching, where if you have a candidate gene, maybe it's a de novo variant in a gene, it's constrained for loss of function variation, but there's no other cases in the world. The gene's never been implicated in disease. That's a nice candidate, but it's certainly not enough evidence to implicate it. But if you could put that in a platform and find out if anyone else has a candidate in the same gene, having found this, a variant of their own in that gene, you just simply be brought together and then see if your phenotypes are in alignment. And I just included this graph from the Orphanet database, which drives home an important point. So they are showing the number of individuals with rare disease based on the frequency or prevalence of each disorder. And what this is showing you is that 80% of the people walking around with rare disease, or some don't walk, but have rare disease, most are due to a small number of common rare diseases, things you've probably heard about: sickle cell anemia, cystic fibrosis, some of these. But most rare diseases are incredibly rare, fewer than 1 in a million, which means no single researcher or clinician will ever have more than one case. So you can't just sit around waiting for the next cases to come and be recruited. The only way to solve these is to share data globally and find that other person around the side of the world that has the same candidate. And that is what we built in the Matchmaker Exchange. This now has 9 different nodes. And so no one is actually sharing their data physically, transferring it. It's all built on a federated approach. And it's one of the first federated genomic data sharing platforms. We worked with Global Alliance to develop standards that all of the databases would use, a common API where all of these can communicate. So our Seeker database is one node. We've submitted 1,502 unique genes over the last 8 years or so. We've received requests, over 500,000 match requests from other users that has actually triggered 13,000 matches. This is what has led to over 300 novel gene discoveries. Now, every match is not a valid match. Sometimes you're like, we got a match. Your patient has a congenital heart defect and mine has hearing loss. Oops, that's not a good match. Move on, but that's okay. A lot of them are good matches, and almost every discovery has benefited from matches on Matchmaker Exchange in our hands. And you don't have to have your own node to hook up to this. We have many nodes in many countries, but most groups actually just log into GeneMatcher, create their own account, and submit their gene there. And that's really easy. And you can see this is just an enormous database with over 15,000 candidates from over 200,000 individuals from 13,000 contributors from 103 countries. I would argue it's the single most useful tool in all of rare disease gene discovery. It's been yielding fruit for many years. However, sometimes you put your candidate in and you get no match. And we need to start to make broader queries of genomic data in order to find these matches, because sometimes people just don't notice this variant that's looking intriguing in that gene. So what I just described is what we also call two-sided matching. So you both have the same candidate and we're just bringing you together. It's actually pretty simple. But what if you don't match anything and you just want to query everybody's genomic data and see if they have this variant you're interested in? It's not in ClinVar, it's not common in Nomad, or it's not in Nomad, but somebody out there probably has this variant and you just want to query all their genomic data sets. That is what we're now focused on. Someday we'll have all of the genomic data queryable in easy ways. That's going to be a while. But we're focused on this variant level matching, a specific query to find genomic data sets. And by way of an example, the same group that launched GeneMatcher have launched Variant Matcher. This is the group at Hopkins led by Nara Sabrera. And this is me making an account and then just putting in a particular variant from a patient that had hypotona, microcephaly, global developmental delay, and esotropia. And when I put that variant in, it hit 8 of individuals in their database. 4 had phenotypes that didn't match mine, and 4 were healthy individuals. They were parents of rare disease proband. So I quickly ruled out this variant that I had no other data, that was not in Nomad, not in ClinVar. But through this analysis of those genomic data sets, found people with a variant and got their phenotypes and could quickly rule that variant out. So this just shows some of the utility. Now, what would be annoying is if you had to one by one search 1,000 databases around the world, right? Everybody's individual data set. That would be a lot of work. So we're trying to overcome that barrier. We're getting all of the groups that have rare disease data sets and genomic data and allow query of them. So these are some of the groups we're collaborating with. This is a project run within the Global Alliance where we're powering it by Global Alliance standards, including the Beacon version 2 API. This allows more complexity to these queries. So instead of just saying, do you have my specific variant, you could also say things like, do you have a case that has a loss of function variant in this gene? Because often when you're finding productive matches, it's not the same variant. So these are kinds of queries you could do and find these cases, even though nobody identified that as a candidate in their data set. So we've now launched the first connection between Variant Matcher and Franklin. Franklin is a commercial platform from Genomic company. And now, just to show you an example, when you're in the Variant Matcher interface and you put in your variant, it automatically will search Franklin while you're doing the search of Variant Matcher. Likewise, when you're in the Franklin platform, which actually is free to use for certain features like the variant search and data sharing, here's one example of searching this variant, and you'll see it right there shows you variant does not appear on Variant Matcher. Or for this other variant submitted 726 times, 144 of which were homozygous on Variant Matcher. And you can click on this URL and it will then land you in Variant Matcher, and then you can get the phenotypes after you log in. So since this was launched in January 2024, Variant Matcher is a much smaller data set. They have run 362 queries from users of their data set. On the other hand, Franklin is a massive commercial platform. They've run 1.3 million queries that have gone back to Variant Matcher. And that's led to 6,824 matches and finding of patients with phenotypes. So it really shows you with just 2 connections, you're already seeing some of the power of this kind of data sharing. So we're continuing to meet and bring other groups onto this platform. Another way we are working is to expand Nomad. So we recently launched V4 that added another 600,000 genomic data sets in the V4 side. Annoyingly, a lot of the added data was of European ancestry, and that's in part because we included the UK Biobank data, which is almost all European ancestry. But I want to show you an interesting observation, which should be obvious, but in the sample growth from V2 to V4, where we added this massive additional European ancestry. And if we look over on the right, the number of variants with allele frequency over 0.1%, which largely means you can filter them out as not being Mendelian causes of disease. What you see is, despite this massive addition of European data, the number of variants with this higher allele frequency almost didn't change at all. Yet with just an incremental increase in non-European data, we saw a much more substantial increase in the number of variants over an allele frequency of 0.1%. What does this say? Basically says we've largely saturated our understanding of common variation in European populations, but we was absolutely not saturated our understanding of common variation in non-European individuals. So it really underscores the importance of getting non-European data into these data sets so that we can filter out common variation that is in various populations and aid our analyses and not be distracted by all of these variants that we could easily filter out if we had better access to diverse data sets. So we are working to bring in both the very diverse All of Us data set. The next version of Nomad will include over 415,000 whole genome sequences from All of Us. And then we're starting a new project in partnership with Global Alliance to build a federated Nomad. And the idea here is there's a lot of data out there that can never be sent to the Broad Institute where we process all the data and put it in Nomad. But because there's national laws that don't allow that data to be exported out of the countries of many places. So we are now going to be working with all of these countries and probably many more to guide them in processing their data in the same way we process and QC the Nomad data, and then have them just send us the final aggregate allele frequencies, which is no longer individual level data. We can then merge that with the Nomad data set and produce a much more diverse set of allele frequencies. And that is a project that we just kicked off in the last few months and are working with a number of groups. And as I said, we'll be adding more. So that will be great. So what I've just talked about is, how we deal with VUSs in novel genes. We're working on building resources to deal with these in known genes, both in Nomad and the VLM platform. We also, I don't have time to talk about the detailed functional studies that we sometimes do, particularly in partnership with Mike Talkowski's group. and many of our collaborators who individually validate variants that are interesting candidates. That takes a lot more work than some of these larger scale approaches. But with that, I'm just going to finish up in the last few minutes and talk a little bit about more genomic medicine implementation. I have a small but mighty team at Mass General Hospital that's really been working to engage stakeholders, identify pain points in delivery of genomic medicine, and deliver solutions. So I also work with representatives of every division and department in the hospital that has anything to do with genetics. So you can see most of the different clinical divisions there. So we have a physician lead, and if they have genetic counselors, a genetic counseling lead. And we query these groups. What's your biggest pain point, like Epic integration or support for billing and insurance inquiries, things like that. Here's the ranking of a past vote, and that kind of guides where we focus our effort. Here's just an example. We help support the renal clinic in launching use of genomics, and they now have 54% of the cases that they're attesting have clinically useful results. Just as an example, this was a 48-year-old female with chronic kidney disease and hypertension, and they were able to identify a genetic cause, which both guided the patient being treatment, as well as identified that her sister can be cleared for kidney donation, which is actually a huge issue in renal diseases, where family members want to donate a kidney to deal with the chronic kidney failure. But if you don't know what the cause is, and that sibling has a 50% chance of having the same disease, you don't really want to put that kidney in. So we also launched a preventive genomics clinic to deal with some high volume needs in our hospital, both hereditary cancer screening for at-risk individuals who meet NCCN guidelines for cancer risk, as well as preconception carrier screening, and dealing with the, as our physicians say, the annoying patients who show up with their direct-to-consumer tests and want us to interpret them. So we launched this clinic. It really worked out well, but we only had 2 physicians covering it. We couldn't scale it to meet the demands of the hospital. So we actually discontinued that clinic, and we have focused on both an e-consult service that we run through the Epic interface, where physicians can ask us questions and we can give answers more quickly and guide them. And we've, I'm just speeding a bit so we have time for questions. That has enabled a lot of output, and the physicians are following our guidance. We just published a paper on that last year. And then my very last slide, what we've really now focused on is partnering genetic counseling services with primary care, because a lot of patients, particularly underrepresented individuals, never make it to specialty referrals. And so if we can pair our GCs with the primary care physicians and help pre-consent and order the genetic testing for them under their order, it really is increasing the number of patients who are getting access to things like basic things like cancer screening and preconception carrier screening. So we're now on a large ramp up with these services, with very expanding across primary care clinics at Mass General Hospital to really improve access to genetics. So with that, I'm going to stop. Thank a lot of people in my team that I work with and a lot of the projects, some of which I talked about today. As I mentioned earlier, if you want a copy of these slides, you can download them from that QR code or this URL. And I'm happy to take any last questions. Thank you.
Thank you so much, Heidi. Wonderful talk, and it's really great to see the impact in the clinic. So we'll take a few questions. Please come to the microphone, or I'll bring you the microphone.
Hi, Heidi. That was a great talk. Earlier in the conference, one of the keynotes was presenting on the pan-genome, and I don't know if you have it reevaluated using the broader pan-genome reference.
That is a planned project within the Greger Consortium. Our Center for Mendelian Genomics is part of a consortium called Greger of 5 different sites, and we're thinking about seeing if there's any added yield from using alternative reference sequences. But I can tell you that the pan-genome references are really not in a state for people to use right now. And so I think it's going to take some time to really develop these alternative references and be able to have them, formatted in a way. They're using graph genomes, which is a technology that already our community has spent 9 years now since Build 37 switched to build 38, and most clinical labs have still not migrated to build 38. So I was just having a conversation earlier about this challenge. So my guess is that other than some research sites that really figure out how to use these and do some analyses, I think it's going to require the sequencing vendors to incorporate these into their platforms. I think this is what's going to actually get these out there in use. And I know Dragon in the Illumina platform has been incorporating graph, reference genome modeling in their platform. So I anticipate the way this is going to be more adopted is relying on the vendors to push it out in their variant calling algorithms. So it makes it much easier for the downstream users to adopt. But so the short answer is no, we haven't yet started working with these, but I think that's how it's going to play out. Yes, Alex.
Hi, Heidi. I liked your slide about the various ways that you can increase yield by doing different technologies. And you mentioned you got 17% yield just by reanalyzing cases.
Yep.
And you mentioned this could be due to advances in gene curation, in variant submissions to ClinVar, and then presumably also advances in tools that variant scientists use to search databases, as well as perhaps who was doing it and how busy they were and what they were able to look at and all that sort of stuff. Do you have a sense about what components of all of those different additive elements is actually driving most of that yield?
Yeah, we haven't done a good breakdown to say how many were new genes that have since been discovered, and then it focused us to look at that gene and any variants in it. I think it's probably just anecdotally been, I'm guessing it's probably a 25% new genes, 75% paying attention to variants that well, actually the RNU4 was a new gene. We just made a discovery last week that was glute, I forget the name of the gene, glute something, where it was an odd start codon modifier. It actually ends up stabilizing the protein that has a higher enzyme efficiency. And then this creates an altered dynamic. And this group ended up having 8 cases with the same variant that affected the use of the start codon and translated to an alternative downstream start codon. The gene had been implicated in a recessive disease, but not a dominant disorder. We had actually seen a de novo variant in the UTR, but had no idea that this very complex mechanism of how it's altering the start codon usage, getting rid of a Dagon sequence that then leads to a change in stabilization of the RNA. So these are complex things that, so the team had seen this de novo. We look at all de novos, those are very suspicious, but it was, a known recessive gene. We only had one variant and we quickly sort of put it aside. And then this paper came out for that gene with a novel mechanism. We re-looked at it, and then we actually through RNA-seq showed the skipping and validated. But I think a lot of it, I think, is a little more on the understanding these variants and what they're actually doing, more so than perhaps a novel gene discovery, although those are a good portion as well. But yeah, we haven't done a systematic look at it though. Yes, I'll be short.
So I will have only one question. So thank you, that was impressive. My question is that you mentioned it quickly about modifier and degenisms. Your thoughts on that?
Modifiers. What was the second part? Modifier and
degenisms, when you have more than one gene that could be, because one of the...
digenic, yes.
Yeah, digenic diseases.
Yeah, I mean, there's been lots of claims of digenic inheritance over the years. Most of them have not been enough evidence, in my opinion, but there are some rare examples where there absolutely is a digenic mechanism. I think what this is going to require is better data sharing, because already the single gene contribution, the primary gene, are so rare that then finding modifiers that you have sufficient evidence to show modifier of a rare disease is just going to require an enormous amount of data sharing to be able to get enough evidence for a lot of these types of examples. And I think this variant level matching platform, where we have access to raw genomic data and you can do more complex queries as are being built in this Beacon API version 2, where we can say things like, find me every case that has a pathogenic variant in this gene and then also has a rare missense variant somewhere else in the gene or in one of these other genes that are in the same pathway. Just show me any cases with a missense variant that's under allele frequency X. These kinds of queries of genomic data around the world, particularly of rare disease cohorts, might be ways that we can find these cases, which are just hard to find enough to really mount a convincing argument of diagenic inheritance. So I think there's a lot to be found there, but it's going to require really hooking up large genomic data sets of rare disease cases to find them. So sorry, that wasn't short enough, but
Wonderful.
thank you very much.
Thank you so much. Let's